Well, the FELIX trial is a sort of a global phase 2 study of Obe-cel, which is our fast off-rate CD19 CAR T-cell product that we've been using for a range of different indications, but on the FELIX study specifically we've been using it for adult acute lymphoblastic leukemia. And so the FELIX study is a sort of a natural extension of what was previously the ALLCAR19 study. That was our phase 1 study that we conducted at UCL, where we saw really promising results in terms of low levels of immunotoxicity, a nice long persistence of the CAR T-cell in the blood, which was associated with durable remissions. So it was really on the strength of that data that the FELIX study was born, if you will, and it's run under Autoleucel Therapeutics. They've been sponsoring the trial, and it's been run in the US, in Spain, and across the UK. And so this is the sort of the top level or the top line results from the FELIX study, early data. But to date, we've actually infused 94 patients on this global study. And I think you're already familiar with the trial design here, but essentially this is an autologous CD19 CAR T-cell therapy. So the patients are leukapheresed to get the starting material for the manufacture. They're then admitted for lymphodepletion with fludarabine and cyclophosphamide. And then there's a couple of safety features with regards to the CAR T-cell infusion. First of all, we split the dose. So we give dose 1 on day 1 and dose 2 on day 10. And we also titrate that day 1 dose according to the bone marrow disease burden that the patient has before their lymphodepletion. So those are two features to try to sort of help the safety profile of the product. So essentially these 94 patients are infused. Notably, 94% of them received both infusions, which is great. And the median duration of follow-up we have on these infused patients is currently 9.5 months. I think when we look at the demographics of this patient population, you have to appreciate that these were a really high-risk group. There were a lot of patients who had very high disease burden prior to lymphodepletion, and there was a significant proportion also had extramedullary disease, and I think we all appreciate that represents a sort of a particularly resistant and difficult phenotype to deal with. All that being said, despite the high burden, despite the circulating blasts, we were actually really successful in terms of our manufacture process. So 94% of the patients who were leukapheresed had a product released for infusion, which was great, and the products were beautifully consistent. The robust manufacturing process ensured that these products were, sort of pretty reproducible, irrespective of baseline disease burden. And in terms of responses, which is of course what we're all interested in, despite this kind of poor risk, patient group, 76% of them achieved complete responses, which again was great. And within those responses, essentially they were really deep responses. So 97% of those were MRD negative responses. So again, very encouraging early data. If we look at those patients, we follow them out, a lot of those responses were durable. So 61% were in ongoing response at a median follow-up of 9.5 months. So again, heartening to see, but we do obviously need extended data to know the true durability of these responses. And what's probably most important for this sort of older comorbid patient population is the fact that this was extremely well tolerated. The toxicity profile was just, the levels were super low. So only 3% of our patients got a grade 3 or more CRS, and only 7% got a grade 3 ICANS. So what this is telling us is that even though we've got patients with lots and lots of disease, the design of this CAR, the fast off-rate binding domain, confers this lesser toxicity profile. Of course, that's exactly what we want to hear as adult hematologists dealing with these frail patients with BALL. So that was a really sort of notable and important piece of our study. And then of course when we look in the peripheral blood, we're seeing beautiful CAR T expansion within that first 28 days, really nice peak and lovely sort of persistence. We're seeing it sort of leveling out at around about 1,000 copies of CAR per microgram of genomic DNA, 6 months, 9 months, 12 months. So this again is reassuring to me, particularly because what I'm seeing is mirroring my experience with ALLCAR19. So I'm sort of hopeful that the FELIX study will be able to recapitulate what we saw in the phase 1 study. So that's a kind of a summary of where we're at with FELIX. It's obviously top line results, it's early days and so on, but so far we've got incredibly encouraging data, and we look forward to sharing more as the months go by at future conferences.